Hey everyone, and welcome to Fact Universe. We hope you're doing great. This is the podcast where we dive into facts and information available in the public domain sourced from credible media outlets and other trusted sources. Let's get started. Today we're embarking on a deep dive into an incredibly promising discovery. Something that could genuinely reshape how we approach cancer therapy. Imagine, just for a moment, a humble creature from the sea holding a key to a powerful new treatment. Sounds almost like science fiction, doesn't it? But scientists from the University of Mississippi have pinpointed a naturally occurring sugar found in sea cucumbers, believe it or not, and it shows genuine cancer-fighting potential. So our mission in this deep dive is to truly unpack what this finding means for you, how this natural compound works its magic, and, well, why it's considered such a significant step forward in the ongoing fight against cancer. We'll be focusing specifically on the sea cucumber species Holothuria floridana and the compound it produces. It's called Fucosylated chondroitin sulfate, or HfFucCS for short. Quite a mouthful. Okay, so let's unpack this a bit.
To really grasp the importance of this discovery, we first need to understand the molecular enemy it targets. Sulfatose-2, or SULF2. Now that's a name you might not have heard before, but apparently it's a significant player in the complex world of cancer. So what exactly is SULF2 and why is it such a critical component in cancer spread? Can you walk us through that?
Absolutely, and it's fascinating, really, just how fundamental SULF2 is to the mechanics of cancer. This enzyme, SULF2, it plays a major role not just in how cancer cells grow, but crucially in how they manage to spread throughout the body, metastasis, basically. You see, our cells, they're normally coated with these complex sugar structures called glycans. Think of them like a dense microscopic forest on the cell's surface. These glycans are vital. They act as the cell's identification tags, like little flags, and also as communication antennas. They regulate all sorts of normal cell behaviors. But cancer, being the incredibly adaptable adversary it is, often hijacks this whole system, and that's where SULF2 steps in. It essentially acts like a molecular gardener, as you mentioned. It starts trimming and rearranging this glycan forest on the cancer cell surface. And it's not random trimming. These are very specific modifications that allow cancer cells to become more aggressive, to move more freely, and even to become more resistant to existing treatments. So the enzyme is, in essence, kind of creating these pathways for cancer to expand and evade detection by the immune system. So yeah, understanding this enzyme, SULF2, is really key to understanding how we might fight cancer spread more effectively.
That molecular gardener analogy really helps visualize it. So it's actively landscaping the cell surface to help the cancer thrive. Okay, so we have this enzyme, SULF2, busily working, reshaping the cellular landscape for cancer's benefit. Now, this is where it gets really interesting. How does this naturally occurring sugar from the sea cucumber, this HfFucCS, actually counteract SULF2? What's the specific mechanism? How does it disrupt this gardening process?
Well, the mechanism is actually quite elegant. It's very direct, very effective. Researchers discovered that HfFucCS, extracted from Holothuria floridana, has this unique ability. It can correctly bind to SULF2, and it's not just a casual interaction. It's a specific binding that effectively blocks SULF2's function. Imagine like a lock and key. HfFucCS acts like a specialized key that gets jammed in the lock, which prevents the real key, the enzyme's normal function, from turning. It basically gums up the works. So by binding to SULF2, HfFucCS disrupts the enzyme's ability to manipulate those crucial glycans, those cellular sugar tags we were talking about. And when SULF2 can't do its gardening, it potentially halts several critical behaviors of cancer cells. This means interfering with their ability to communicate with each other, hindering their movement, which is essential for metastasis, the spread of cancer. And it might even impact their capacity to evade the body's immune system. So it's not just a subtle effect, it's quite a direct intervention that could severely cripple a cancer cell's ability to thrive and, importantly, to spread.
Wow, that sounds like a remarkably precise counterattack, hitting it right where it hurts, at a fundamental level. But, with any major scientific breakthrough like this, a crucial question always comes up. How confident are the scientists in these findings? Was this simply a theoretical discovery, maybe something seen in a computer model, or was it thoroughly tested and validated in the lab? And beyond just the science, what makes this specific compound, HfFucCS, uniquely promising compared to other potential treatments? Are there practical advantages that really set it apart?
Yeah, that's a vital line of inquiry. And the confidence in these findings is actually quite high. It's high because the compound went through a rigorous two-pronged validation approach. They didn't just rely on one method. They used both advanced computational modeling and real hands-on lab experiments, which is key. The computational simulations, they predicted precisely how HfFucCS would interact with SULF2, like creating a molecular blueprint of the binding. Then, and this is the crucial part, or laboratory assays, actual wet lab experiments confirm those predictions. They demonstrated that the sugar effectively binds to and inhibits the enzyme, just as the models suggested it would. This consistency, between the prediction and the empirical proof, it gives researchers a very strong degree of confidence in its potential. Now, regarding its practical advantages, there's a truly crucial added bonus that sets HfFucCS apart. And this is a big one. It doesn't seem to interfere with blood clotting.
Okay, why is that so important?
Well, it's a massive deal in drug development. You see, many other compounds that have been investigated to target SULF2, while they might be effective against the cancer aspect, they often come with a significant side effect. They can increase the risk of bleeding, and that obviously makes them less ideal for clinical use, right? Especially for patients who might already be frail or undergoing other treatments. HfFucCS appears to bypass this critical safety concern entirely, which could make it a much safer alternative. And there's another thing. In the broader context of drug production, there's a significant advantage in its source. Traditionally, similar complex sugar compounds, like chondroitin sulfate you might find in supplements, are often extracted from mammalian tissues, like cow cartilage or something. But that comes with inherent risks. Contamination risks, like the potential for viruses or prions.
Right, things you definitely don't want in a medicine.
Exactly. Marine sources, like the sea cucumber, are generally considered far less prone to these types of contamination risks. So this points towards a potentially cleaner, maybe more sustainable route for producing these kinds of complex molecules. It's safer and possibly easier to standardize.
So these advantages, together with the safety profile, the cleaner source, they really paint a picture of a potentially much more accessible future treatment if it pans out. Okay, those are indeed significant plus points. It really underscores the potential impact here. It's not just a new potential drug, but maybe a new way of finding and making drugs. But, as we know, the journey from a lab discovery, however exciting, to actually helping patients, that's often a long road, filled with hurdles. So what does this all mean for getting HfFucCS from the lab bench to the bedside? What hurdles still remain? And what are the immediate next steps in the research process? Where do they go from here?
That's the million-dollar question, isn't it? And it brings us straight to one of the primary challenges: supply. It's a practical hurdle. While sea cucumbers are fascinating creatures, they simply aren't abundant enough in the wild to meet the kind of massive pharmaceutical-scale demand we need for a widely available drug. You can't just go out and harvest millions of them from the ocean floor. It's not sustainable, and you wouldn't get a consistent product either.
Right, the environmental impact alone would be huge, not to mention the variability.
Precisely. So the solution the research team is actively pursuing now is chemical synthesis. They're working on essentially recreating HfFucCS from scratch in the lab, building it molecule by molecule. This allows for much larger scale production, and importantly, it ensures a consistent, pure, and safe formulation that can be used for more extensive testing. And ultimately, if it gets that far for clinical use, it's about replicating nature's blueprint, but in a controlled, scalable way. So the immediate next steps are absolutely crucial. Once they have enough of this lab-synthesized compound, it will be rigorously tested in animal models, preclinical testing. This is where they'll evaluate its effectiveness, does it actually work against cancer in a living system, and critically, its safety profile over time, looking for any unexpected side effects in a whole organism. If, and it's still an if at this stage, but if it proves successful and safe in those animal trials, then and only then could it potentially move forward into human clinical trials, phase 1, phase 2, and so on. It's a meticulous process, step by step, but that's how a new therapeutic weapon slowly but surely makes its way towards helping patients.
It's clear that scaling up is a major hurdle. Yeah, but that synthesis plan sounds really promising. It's a great example of scientific ingenuity finding ways around natural limitations. Something else that really stood out, reading about this research, was how interdisciplinary it was. It wasn't just one type of scientist working in isolation in their own little silo. How crucial was that collaboration between different scientific fields in actually making this breakthrough happen? It really seems like this kind of complex problem demands a truly diverse team effort, doesn't it?
Oh, absolutely. You've hit on a really critical point there. This discovery is a fantastic example of the immense power of interdisciplinary science. It genuinely is. The team behind this study wasn't just, say, marine biologists or just chemists. It intentionally brought together experts with deep knowledge in biomolecular sciences, pharmacology, medicinal chemistry, and computational biology. And each of these fields brings a completely unique perspective and, crucially, a unique set of tools to the table. So, for instance, the biomolecular scientists, they understood the intricate structures of the glycans and SOLF2 at that fundamental molecular level. Meanwhile, the computational biologists could actually model those interactions virtually. They could predict outcomes, test hypotheses in the computer before anyone even picked up a test tube.
Saving time and resources, presumably.
Exactly. And then the medicinal chemists were vital for figuring out how to potentially synthesize the compound, how to refine it, maybe make it more potent or stable, turning it into a viable drug candidate. And the pharmacologists, of course, focused on how it would actually behave in a biological system, its absorption, distribution, potential toxicity, its therapeutic potential overall. Their collective expertise allowed them not just to discover HfFucCS and figure out what it was, but also to thoroughly evaluate its function, how it worked, and importantly, to start planning for its future as a potential drug. It really reinforces this idea that the most challenging scientific puzzles, especially complex ones related to human health like cancer, often demand this kind of collaborative, multifaceted approach to truly unlock their secrets. You need all those different angles.
What an incredible journey we've taken today in this deep dive. Seriously, from the mysterious depths of the ocean, finding this compound in a sea cucumber, all the way to the cutting edge of cancer research, understanding these intricate molecular mechanisms. We've explored this surprising discovery from the natural world. We've delved into how it targets cancer at that fundamental molecular level by inhibiting SOLF2, and we've looked at its promising future while also acknowledging the very real challenges, like synthesis and testing, that still lie ahead. So as we wrap up, considering the immense potential packed into this humble sea cucumber, providing a potential key to fighting a disease as complex as cancer, it really does raise an important, maybe even profound question for all of us to think about. What other secrets might nature hold for our health? What other solutions are out there, in oceans, forests, maybe even our own backyards, just waiting for curious minds to uncover them? That wraps up today's episode of Fact Universe. What are your thoughts on what we discussed today? We'd love to hear your opinions. Thanks so much for tuning in. We hope you enjoyed it. Your feedback means a lot and helps us keep improving. Don't forget to like, share, and subscribe to support the podcast and help us grow. See you next time.